The manufacture of a drug is not a simple process and easy as many think. The development of a single drug requires a series of steps, which makes it a very long and expensive process. In this video we talk about the first and second stage of this process. The design strategy for drug discovery begins with knowledge of the genetic and molecular basis of disease and the use of that information to select a specific therapeutic target. Then are designed drugs to interact with the target. Through rational design aims to search a drug that is highly specific for a particular target in a disease with the hope of achieving a better therapeutic outcome without adverse reactions. The pre-discovery stage turn involves several processes. The pharmaceutical company seeks to answer questions like which diseases representing an important medical need in humans of a community, if there is or not a treatment for the relief of the disease. If the answer is yes, then the company proceeds to conducting a research program to better understand the disease. Each new drug is the result of an intensive search process during which examines all possibilities to develop it. Therefore, steps to develop new molecules and drugs requires a large investment of time, money and resources. Once the company selected the disease for which work, the next step is to determine the potential therapeutic targets or objective on which it will act the candidate molecules. It is important to mention that a therapeutic target or objective is a molecule that plays a key role in a disease. That is the place where the drug exerts its action. When the lenses inside the cell can be used, only can use drugs that cross into the cell membrane. It is important to consider the various causes of diseases as whether it is hereditary or is caused by an external pathogen, either a virus or bacteria. The purpose of discovery is to identify the targets of these causes and develop drugs that act on these, blocking or enhancing the target. This can be achieved by various technological methods such as DNA chip experiments, protein electrophoresis, mass spectrometry, DNA sequencing and computer imaging techniques. Thus, today there are about 8,000 known therapeutic targets. Once the therapeutic target identified, the next step is to validate it. The purpose of the validation is actually demonstrate that the molecule is involved in the disease and it confirm if it is a candidate. Validation is the target used in healthy tissues to trigger the disease and then lock it to restore the healthy condition. Among the many treatments or therapeutic targets receivers are cancer treatment-based chemotherapy with cytotoxic substances that suppress cell growth of both tumor cells and healthy cells. The progress of the biotechnology has allowed it to find receptors such as the receptor tyrosine kinase, which possesses the ability to catalyze the transfer of phosphate groups and the molecule of ATP to hydroxyl groups, those tyrosine that may be present in the receptors themselves, as in particular capable of being phosphorylated by them. Is a cell receptor associated with a signaling pathway and belongs to the family of receptor with intrinsic enzymatic activity, are associated and has as ligands to insulin, epidermal growth factor, the fibroblast growth factor, neurotrophin, and the other factors trophic. Another particular example is the influenza viral disease, where the peak cell receptor binds to the hemagglutinin HA protein. Less than tau G equals 1 duration equals 0/ greater than. Be aware that in the case of influenza viruses, replication is limited to the respiratory tract as maturation of the viral particle requires the action of proteases. The proteases employing the viral particle are serine proteases secreted by clear cells that allow the cleavage of HA0 and HA1, and HA2. The formation of these two peptides is required for fusion of the viral membrane with vacuole. Acetophilus. Previously mentioned that the search for targets using various technological methods. We will discuss 3 of these methods are tests in vitro and in vivo. heights and NMR screening. In vitro tests are performed on isolated cells, tissues, receptors or enzymes and commonly are performed before testing in vivo because they are cheaper than these, less controversial and easier to perform, mainly because it can be automated and is no longer isolating and modifying enzymes problem to realize. In the case of in vivo tests using transgenic animals, its genetic code has been altered, which are treated with the drug to see if alleviates the problem by eliminating the visible symptoms. The disadvantages of this type of tests is that they are slow, cause suffering to animals, can cause problems in pharmacokinetics, resulting in erroneous results or difficult to interpret. Thus, the in vitro tests are used to determine drug receptor interactions and to identify the in vivo pharmacokinetic properties. The disadvantages of this type of tests is that they are slow, cause suffering to animals, can cause problems in pharmacokinetics, causing erroneous results or difficult to interpret. Thus, the in vitro tests are used to determine drug receptor interactions and to identify the in vivo pharmacokinetic properties. The automation of tests can be done by techniques such as heights, which can perform up to 50 biochemical tests. These tests are based reactions perform easily quantifiable as cell growth, enzymatic reactions which are displayed in a color change or with radioactively labeled ligand displacement. The screening is done to observe that in the tests are compounds that inhibit binding between a compound and the radiolabeled ligand, thus, to identify whether a compound binds to a target using methods such as NMR spectroscopy, which is an analytical tool where a compound is subjected to radiation with a short pulse of energy and thus the nuclei of hydrogen atoms, carbon or nitrogen are excited, and when radiation is just the core excitation decays, slowly relaxing and releasing energy. The molecules are identified by their relaxation times, which vary depending on the medium or position of each atom in the molecule. Drugs that are small molecules typically have longer relaxation times, and proteins are large molecules that have a short relaxation time. Is therefore possible to detect the binding between a protein and a test compound in this case as the drug is designed. As you can see, the start for the manufacture of a drug involves many important steps which should be taken with the utmost seriousness and importance, being as each one of the steps to follow can significantly influence in the further drug development successfully in the following steps to get a drug that hits the market and is available to those who require it.